Posture is defined as a composite of all joint positions of the body at a given moment. An erect posture, where an individual is standing straight up, is ideal when a line can be drawn vertically through the body's center of mass to the ground. Faulty balance, described as postural sway, consists of constant displacement and repositioning of the body's center of gravity. The calf muscles soleus and gastrocnemius connect the Achilles tendon to the back of the lower leg bones and the back of the knee. Calf muscles are actively involved in the maintenance of balance via paradoxical contractions, for which muscle tension adjusts to move the muscles in the body in opposite directions, thus repositioning the body's center of gravity as needed for stability. In this experiment, contraction of the calf muscles is induced via voltage stimulation to the sural nerve, which runs through the gastrocnemius and soleus muscle region. It is our objective to determine the effects of disturbance to muscle contraction on postural stability during static stance. The voltage disturbance must be below the levels required for eliciting nociceptive responses, as this may cause destabilization due to voluntary contractions. Our hypothesis is that low-level electrical stimulation to the sural nerve will result in muscle contractions that cause postural destabilization during standing. Results of this study could be applied to the development of assistive technologies for individuals without the capacity to generate large active movements, such as those with spinal cord injury, to improve their range of postures or joint properties involved in stabilization. Observations of muscle contraction from electrical stimulation used electrocutaneous stimulation in EMG recordings. This followed methods used in previously conducted studies regarding the effects of electrical stimulation on muscle movement. EMG recordings were used to monitor muscle contraction pre, during, and post-stimulation as indication of perturbance. Differential EMG electrodes were placed over the gastrocnemius muscle of the right leg, and the reference electrode was attached over the lateral epicondyle of the femur. To elicit muscle contractions, electrical stimulations were applied to the retromalleolar pathway of the sural nerve on the lateral side of the right leg proximal to the ankle bone. Stimulations were in the range of those used in previous studies to elicit muscle movement without eliciting a nociceptive flexion reflex that would induce instability from voluntary movement. Two different voltages, 23 and 50 volts, were used to determine whether increased strength of muscle contraction resulted in increased instability. Stability was measured using center of pressure analysis with the Nintendo Wii Fit board. The center of pressures, pre, during, and post-stimulation for both voltages were recorded with a LabVIEW program. Instability was defined as increased average distance of individual center of pressure measurements from the reference point of the entire center of pressure data. All subjects were asked to assume a static stance with their feet placed together and arms crossed so that the stance could be easily replicated. In between stimulations, subjects assumed a rest-sitting position in order to prevent muscle adjustment to the stance. All subjects were selected from a group of students. The sole criteria for rejection for participation was a history of neural or neuromuscular disease or leg injuries. From this method, a sample size of 6 was determined from preliminary data using a power of 95%. The results of the experiment were then analyzed to determine whether or not electrical stimulation of the muscle significantly affects a person's ability to maintain a relatively constant center of pressure. First, a center point of each trial's data was found by averaging the X and Y values output from the Wii board. Next, the average center of pressure distance from this center point was determined for each trial using the distance formula. The analysis involved two paired t-tests with a power of 95%. The first t-test compared the average center of pressure distance from the center point of the zero stimulation test to that of the 23-volt stimulation test. This test yielded a p-value of 0.2846. Thus, the null hypothesis that 23-volt stimulation would not significantly disrupt a person's center of pressure cannot be rejected. The second t-test compared the 0 and 50-volt stimulation test, yielding a p-value of 0.0362. Thus, the null hypothesis of this test can be rejected, indicating that 50-volt stimulation does significantly disrupt center of pressure. To assess the initial hypothesis that electrically induced muscle perturbance would cause postural destabilization during static stance, there were two null hypotheses developed for the two stimulation strengths. Numerically, the average distance from the center position increased with stimulation, and the increase corresponded to the strength of stimulation. There was a statistically insignificant numerical increase in the stability between 0 and 23 volts of stimulation. This implied that 23 volts did not result in destabilization. Stimulation with 50 volts produced a statistically significant difference, thus indicating postural destabilization during static stance. The statistically contradictory differences between the two stimulation strengths corresponded with previous studies that indicated a threshold level of perturbance was required for alteration in postural control. Studies of balance training also indicated that increased strength of stimulation did increase subsensory neural reflex magnitude, and a direct correlation was found between decreased stability and increased reflex magnitude. Additionally, studies of stimulation for muscle activation indicated that stimulations as high as 50 volts did not activate sensory muscles. Thus, destabilization is not a direct cause of voluntary movement. From this, we can conclude that muscle perturbance via electrical stimulation induced postural instability during static stance.